So for this video, we're going to talk about the cell cycle. And when we say the cell cycle, we don't mean a cell on a bicycle riding around France. We really mean the process by which one cell becomes two cells. So this is continuing along with our discussion on cellular division, and the cell cycle is the overlapping concept that's going to describe everything that a cell needs to do to go from one cell to two cells. And when we talk about the cell cycle, we're really going to summarize the cell cycle literally as a circle, as a cycle. And what I want to do is talk you through this cycle so that you have a sort of anchor for what's going on in the cell cycle, and then we'll talk about some more of the specifics as we go along. So let's start right here on the cell cycle at a process known as cytokinesis. And we're only starting here because it's a convenient place to start. Now, as you know, the cycle goes around and around and around, so we could start anywhere in this cycle. But we'll start at cytokinesis because that's the step at which one cell becomes two cells. And once that one cell becomes two cells, each individual cell then starts the process all over again. So if we start with an individual cell, it just was created from a new cell, so we would call this single cell a mother cell. And we call the two cells that come from it a daughter cell. So we're starting with a new daughter cell. The first thing this daughter cell is going to do in the cell cycle is known as the G1 phase. So during the G1 phase, this daughter cell is going to start growing. Because if it wants to divide, the first thing it needs to do is get a little bit bigger. So during the G1 phase, this cell is going to start to grow, it's going to duplicate all of the organelles inside, it's going to make extra copies of everything, so that it's ready to become two cells again. Once it goes through the G1 phase, we're then going to go into the next step known as the S phase. The S phase, S stands for synthesis. So in the S phase, that's where the cell is going to duplicate all of its DNA. And we'll talk about, when you watch the lecture on mitosis, why it's important that the DNA is duplicated. But you can easily understand that for one cell to become two cells, you need twice as much DNA. So the S phase is where the cell grows and duplicates its DNA. So we've taken this daughter cell now, we've grown larger and duplicated all of its organelles in G1. Now we've duplicated all of its DNA in S phase. And our final step is to go to G2 phase, where the cell grows just a little bit bigger. It starts growing some more enzymes that it needs for processing things. It's finishing up getting ready to divide. So G1, S phase, and G2 phase are also called interphase. So we'll call these three steps of the cell cycle interphase. And most of the time, your cell, any cell you look at, is going to be an interphase. That's where it's going through either the process of duplicating its organelles, duplicating its DNA, or just growing the final little bit that it needs to grow. So aside from interphase, the last step left in the cell cycle is known as M phase. And M phase includes two important processes: mitosis, which is the division of the nucleus, so one nucleus into two nuclei, and then cytokinesis, which we've already discussed, is the division of one cell into two cells. So during M phase, that's where all of the magic happens with the cell cycle. That's where one cell actually becomes two. And this process of mitosis is so important to understand that we'll cover that in another lecture. But for now, just know that we go through interphase, G1, S phase, G2, and then we get into M phase, where we do mitosis and cytokinesis. And this cycle goes around and around and around. So one cell going through the cell cycle will then give us two cells. Two cells going through the cell cycle will then give us four cells, and so on and so forth. Now there's one thing that we didn't talk about in that last diagram, and that's the option of leaving the cell cycle. Now certain cells in your body can actually leave this cycle. They can stop going around MG1, SG2, MG1, SG2. They can stop duplicating, and they do this by going to a stage known as G0. And when a cell is in G0 phase, it's just sitting there being dormant. It's no longer growing, and we call that cell quiescent, or say it's in a quiescence state. Okay, so G0 is an option for these cells. They can leave the cell cycle and then go to G0. Sometimes cells that are in G0 can then return to the cell cycle and grow again. Now there's different types of cells in your body. Each type of cell has a different preference for G0 or not. Just by way of example, your epithelial cells, so the cells that make up your skin, almost never go to G0. And that should be a pretty intuitive thought for you, because your skin is constantly growing. You're constantly growing new skin. You're constantly getting rid of dead skin cells. So those cells never have a chance to take a break. They never stop growing. They're always growing, always dividing. You always need more skin cells. They almost never go to G0. We can compare that to cells in your kidney and your liver, specifically in the parenchyma of your kidney and liver. These cells, when you reach adulthood, stop growing. You stop growing your kidneys, you stop growing your liver. So most of the cells in there will go to G0. They're no longer growing. They're just sitting there doing their job, whatever it is in those organs. However, in some cases, these cells can actually re-enter the cell cycle and start dividing again. And this occurs if you have, for example, injuries to your liver or your kidneys. In some cases, those injuries can be repaired by new cells growing and new cells filling in those damaged areas. Now, this doesn't always occur. They can only sometimes go back into the cell cycle. However, it is a possibility. Now, the last example I want to give you is neurons. And as you all know, if you suffer a spinal cord injury, that's a pretty permanent injury for one to have. And that's because your neurons, once they're done growing, once your neurons throughout your body are done growing, they go to G0, they're quiescent, and they almost never re-enter the cell cycle. Once you have neurons, once you have brain cells, they are not dividing anymore. They're there, that's all you have. If you start losing them, it's really difficult to cause them to re-enter the cell cycle and regrow. And this is where some of the research has gone into using stem cells to repair spinal cord injuries, for example, because those stem cells haven't reached G0 yet. However, that's an entirely different process that's way more complicated, and we're not going to talk about it in this class. So how is this cell cycle regulated? As you can imagine, as the cell goes through the cycle, it goes around and around. I suppose we should go this way. Around and around and around and around. The cell cycle is controlling how the cell divides. And that's important to the cell because that's how you prevent cancer. And we talked about cancer being inappropriate division of the cell. So if the cell cycle is not regulated, if there's not places where the cell stops and accounts for itself, make sure that things are going properly, you're going to end up with cancer. So if we look at this normal cycle here, G1, S, G2, M, with the option of going out to G0, we're going to have three major checkpoints that we're going to talk about. The first checkpoint we have comes during the G1 phase of the cell cycle. And the G1 checkpoint is really asking the question, should the cell divide? So the cell is going to decide whether or not it should divide based on some different parameters. One of these is whether there are growth factors. So cells excrete different chemicals, different hormones that tell the surrounding cells they need to grow. So one question the cell is going to ask during G1 is, do I have growth factors? Are there growth factors around? Are other cells telling me to grow? The other thing that's going to be checked during G1 is whether the cell is large enough. So the important purpose of G1 is to duplicate all the organelles, to grow a cell that's large enough that when you divide it into two, you can still have two complete cells. So the cell is going to check whether or not it's large enough, because if it's not large enough, it's not going to continue on through the process. The last part of this checkpoint is there enough nutrients? Is there enough food around for two cells to eat when there's only one cell? And this is an important question to ask, because if there's not enough nutrients, there's no need to divide into two cells, because you will starve one or both of those cells once you do that. So these are the important questions that G1 asks, and if all of these factors are true, if there are growth factors, if the cell is large enough, and if the cell has enough nutrients, then it will continue on to S phase. If it doesn't have any of these parameters, it's going to sit and it's going to wait in the G1 phase until those parameters are met. And this is part of the reason why cells spend most of their time in interphase, somewhere between G1 and G2, because these checkpoints will hold cells up until everything is true. So let's say all of these things happen. The G1 checkpoints are all met. We continue on to S phase. And you remember that during S phase, that's when the DNA is being synthesized, is being duplicated. Once we get through S phase, we're going to go to the final G2 phase. That's where the cell is going to do its last little bit of growing, but there's also an important checkpoint in G2. And the important checkpoint in G2 is, was all of the DNA replicated okay? And that's the most important question you can have after synthesizing all of this DNA. Did you actually duplicate every piece of DNA that you have, that the original cell had? And are those duplicates reasonably accurate to what the parental versions of those genes were? And the cell has various ways that it can check through this. It has various ways of checking whether there's gross damage to the DNA. But the long story short, if the DNA was not replicated okay, the cell is going to hold up here in the G2 phase. Now sometimes what it's doing is it's holding up and just waiting for some of the processes that started during S phase to finish. And once the DNA is replicated okay, then it will continue on to mitosis, the M phase. The other question that's asked during the G2 checkpoint is the cell large enough again? We asked here whether it was large enough to go on to S phase, but we're going to ask one last time, is it large enough? Is it really large enough to divide into two cells? So assuming you meet all of the G2 checkpoints, then you're going to go into mitosis. And as I mentioned, we're going to talk about mitosis in another video. But there is an important checkpoint during mitosis known as the metaphase checkpoint. And the word metaphase might not mean anything to you until you viewed the video on the mitosis process. But basically, in the metaphase checkpoint, it's asking whether all of the chromosomes are attached to spindles within the cell. Are all the chromosomes attached so that we can divide them into two parts? If they're not all attached, some might be left behind, some might get all dragged to one side and not the other. So this is an important checkpoint that makes sure that everything is attached and everything is going to be divided equally between the two. And so as you go through all of these checkpoints, it's checking that the cell is doing everything properly. If you have any loss of function in any of these checkpoints, you're going to develop inappropriate division, which could lead to cancer in the cells. Now, because this regulation of cancer, this regulation of the cell cycle is so critical to cancer, a lot of research has been done into the genes that regulate the cell cycle. And I'll go ahead and give you a little bit of a teaser here. There are a lot of genes that regulate the cell cycle, and we're not going to talk about all of the genes. We're not even going to talk about any of the specific genes in this class that regulate the cell cycle. But I do want you to know that there are two major classes of genes that can be thought of to regulate the cell cycle. One of those is known as a proto-oncogene, which mutates into what scientists call an oncogene, and you've probably heard the term oncogene before. So the normal function of a proto-oncogene is to drive the cell cycle forward when it's appropriate. So the proto-oncogene is a type of protein, is a type of enzyme that says, all right, checkpoints met, let's do X, Y, and Z and start dividing. If you mutate to an oncogene, that mutant function is going to drive the cell cycle forward all the time. So instead of saying, all right, the checkpoint is met, let's divide, this oncogene is going to say, let's divide, let's divide, let's divide, let's divide. I don't care that we're not ready, let's divide. And you can see how that's going to lead to cancer. So that's one type of class of genes that regulate the cell cycle. Another class of genes that regulate the cell cycle are known as tumor suppressor genes. And these mutate to mutant tumor suppressor genes. They don't really have a fancy name like oncogenes. Sometimes people call these anti-oncogenes because they have the opposite function of an oncogene. So the normal function of a tumor suppressor gene, as you can imagine, is to suppress the formation of a tumor, or actually to stop cells from dividing when they shouldn't. So these are the guys, the genes that are in there saying, okay, we're not ready, checkpoint's not met, let's not divide. Now when they mutate, they no longer recognize, for example, that a checkpoint's not met. So they don't stop inappropriate division ever. So once they're mutant, they never stop. So again, you have this process where the checkpoint's not met, but instead of this guy coming in and saying, all right, stop, it doesn't say anything. Okay, so it doesn't say let's stop. The cell keeps dividing, keeps dividing, keeps dividing, keeps dividing, keeps dividing. You can see how that leads to cancer. There's a couple other ways that we can describe these things. The proto-oncogenes can be considered an accelerator. So this is like a gas pedal in a car. If you have a gas pedal, you can push forward to accelerate the car. If that gas pedal is broken, it's going to push all the time. That car is going to go forward, it's going to crash. We're going to call that crash cancer. All right, the tumor suppressor gene, as opposed to the accelerator, can be considered the brake. So the normal function of the tumor suppressor gene is that you can hit the brake, you can stop the cell cycle from moving forward. However, if that tumor suppressor gene is mutated, you no longer have a brake. So even when you see mistakes, even when the checkpoints recognize that there are mistakes, you no longer have the ability to stop that cell from driving forward. It's going to keep accelerating, moving forward, it's going to then crash, and you have cancer again. So all of these functions are important for regulating the cell cycle, and the numbers of proto-oncogenes and tumor suppressor genes are growing every day. There are lots of different genes involved. These are just two big classes that they're divided into. So the last piece I want to do is just review once again what we talked about. So when we talked about the cell cycle, we talked about a couple phases, starting with cytokinesis here, where we had a brand new daughter cell. We first go into G1 phase, and G1 phase is where the organelles duplicate and the cell grows a little bit. Once we get through the G1 phase and we meet all the requirements of the G1 checkpoint, we then go into S phase. And S phase is where the cell is going to duplicate all of its DNA. It's going to replicate the DNA and prepare it to divide. Once we go through the S phase, we're going to go into the G2 phase. And in the G2 phase, we're going to once again have another checkpoint. Here's the G2 checkpoint. This makes sure that everything occurred properly in the process of the S phase, made sure all the DNA was replicated again. And if that checkpoint is met, then we'll go into the M phase. So we'll go into first mitosis, and then cytokinesis. And you'll remember that mitosis is the division of the nucleus, one nucleus into two nuclei, and cytokinesis is the division of one cell into two cells. And these occur back to back during the M phase. Now, once the cell is done with the M phase, it's going to go back again and start over this cell cycle. However, it does have the option, as you remember, of leaving the cell cycle and going to the G0 phase. And in the G0 phase, the cell is said to be quiescent. And it's going to sit here and not grow any longer. However, in some cases, it can re-enter the cell cycle from the G0 phase. And we talked about some of those specific circumstances. So just to go through it again, G1, S, G2, M, start back over at G1, S, G2, M. We'll call G1 through G2 interphase. And then we have interphase and we have M phase. And you can see just from this diagram that the cell is going to spend most of its time in interphase. So remember this cell cycle process. Watch the other video on mitosis, which will focus really in on the M phase and let you see exactly what's going on in there.